Hello, everyone. My name is Diego Rodriguez. I am a research specialist at the State Monogastric Nutrition Laboratory, and today I will share data about the digestibility values of soybean meal and soybean expeller when fed to growing pigs. Oilseed production around the world had been increased for the last century. About 575 million metric tons of oilseed are produced every year, and about 60% of the total oilseed production are soybean, having Brazil and the United States as the biggest producer with about 50% of the total oilseed production. Also, it's been reported that about 239 million metric tons of soybean meal are produced, and this soybean meal is mainly used in the formulation for diets for monogastric animals, especially the use in poultry diets, but also in swine industry. And that is because of the great source of protein that can be found in soybean meal. There are two mechanisms that are usually used for oil extraction from soybean. Solvent extraction is the mechanism more used, and there is mechanical or expeller extraction. Usually for solvent extraction, they use a chemical named hexane and also use high temperature that facilitate the oil extraction, and therefore the meal fraction from this process has a lower oil content compared with mechanical pressing. Also, there is a concern the use of chemical during the processing of feed ingredients, and therefore the soybean oil that is obtained by mechanical pressing may have a greater value in the food market compared with the soybean oil that is obtained by solvent extraction. It is important also mention that solvent extraction cannot be done in a small scale compared with mechanical extraction, and this is more important for a small producer of soybeans. Knowing this process, Instapro International developed a mechanism that is named high shear dry extrusion. And this process consists that the soybean is exposed to high temperature, high pressure, high shear in a relatively short time, and this high temperature short time process, it is believed that help in the rupture of plant cell walls to facilitate the oil extraction from the soybean, but also because of the high temperature that is used during this process, it may help in the deactivation of antinutritional factors such as trypsin inhibitors. After the high shear dry extrusion process, the meal fraction from this process is mechanical present, and therefore the end product will have a high value soybean oil that can be sold in the food market with a greater price, and also an expelled soybean meal fraction that could be used in the formulation of diets for monogastric animals. Therefore, the objective of this study was to test the hypothesis that including soybean expeller to diets fed to growing pigs will increase the standardized ileal digestibility of amino acid, also the apparent total tract digestibility of gross energy and total dietary fiber, and also an increase of concentration of energy compared with conventional solvent extract soybean meal. To test this hypothesis, we conduct two experiments. The first experiment was to determine the amino acid digestibility, and for this experiment, we utilized three diets. The first experimental diet was a cornstarch with 35% of soybean expeller, and the second was cornstarch and 33% of soybean meal as the only source of amino acid. For the diet with soybean meal, we include 2% of soybean oil, and that is because it is reported that oil has a positive effect on amino acid digestibility. And because soybean expeller has a greater oil content compared with soybean meal, we wanted to have similar oil concentration in both diets to see a better effect on amino acid digestibility. In our second experiment, we formulate three diets: a corn basal diet, plus two additional diets including corn and 32% of soybean expeller, or corn and 30% of soybean meal. For the experimental design, for the amino acid digestibility experiment, we utilized 9 cannulated barrels with an initial body weight of 56 kilograms, and we allowed those pigs in a triplicate 3x3 Latin square design, and therefore we had 9 pigs per diet. For our second experiment, we utilized 24 growing barrels with an initial body weight of 45 kilograms, and these 24 barrels were allotted to the 3 experimental diets by using a randomized complete block design, and therefore we had 8 replicate peaks per diet. For the sample collection, for amino acid digestibility experiment, pigs were fed the experimental diets for a total of 7 days, having the initial 5 days as adaptation period, followed by 2 days of ileal digesta sample collections. And for the energy and nutrient digestibility experiment, pigs were fed the experimental diets for 9 days, having the initial 5 days as adaptation period to the diet, followed by 4 days of total but separate urine and fecal collection. Jumping into the results, first let me set up the slides. In orange color, soybean expeller will be represented, and in dark blue color, soybean meal will be represented. As was expected, acid hydrolyzed ether extract was greater in soybean expeller compared with soybean meal. And this result was expected because of the high oil content that is observed after pressing the soybean compared with solvent extraction. In terms of crude protein, we observed that the values of both soybeans are around 45 to 46%, which is a little bit lower compared with reported values such as the NRC 2012. In terms of fiber, insoluble and soluble dietary fiber were similar concentration between soybean expeller and soybean meal. In terms of gross energy, because of the high acid hydrolyzed ether extract in soybean expeller, it was expected that the gross energy was greater in soybean expeller compared with soybean meal. Trypsin inhibitor is an antinutritional factor that is commonly found in soybean products, and this has a negative effect in amino acid digestibility. A way to ameliorate the negative effect of trypsin inhibitor is by using heat treatments. And after the heat process that is commonly used for soybean expeller and soybean meal, there was a reduction in trypsin inhibitor in both soybean products, having a greater reduction in soybean meal compared with soybean expeller. Also, it's been reported that values close to 4 units per milligram of trypsin inhibitors are enough to have no effects on amino acid digestibility. Therefore, we analyzed the standardized ileal digestibility of amino acid, and we found out that soybean expeller has greater SID of amino acid compared with soybean meal, with the exception of lysine and tryptophan that tended to be greater in soybean expeller compared with soybean meal. This effect is likely because of a positive effect during the dry extrusion process that allow a better utilization of the protein from soybean expeller compared with soybean meal. In terms of apparent total tract digestibility of gross energy, there was no difference among soybean products and corn. However, the apparent total tract digestibility of total dietary fiber was greater in soybean expeller compared with corn. And this effect may be explained because of a greater soluble dietary fiber concentration in soybean products compared with corn. On the other hand, there was no difference in total dietary fiber digestibility between soybean expeller and soybean meal. In terms of energy concentration, soybean expeller has a greater digestible energy compared with soybean meal and corn. And the same was observed for metabolizable energy, where soybean meal and corn has lower ME compared with soybean expeller. The reason of this result is likely because of a greater oil content in soybean expeller compared with soybean meal and corn. And it is known that fat produces more energy and less heat production compared with starch and proteins. Based on the metabolizable energy values and the nutrient composition of the feed ingredients, we calculate the net energy of the feed ingredients using the prediction equation from Noblet. And it was observed that soybean meal has the lower net energy compared with soybean expeller and corn. However, there was no difference between soybean expeller and corn in net energy. Based on the energy values, we calculate the metabolizability of energy to estimate how much of the energy was utilized for the pig. And it was observed that the ME to the E ratio in corn was greater compared with soybean meal. And this result may be explained because of the high concentration of crude protein in soybean meal compared with corn. And it is known that the efficiency of utilization of energy in proteins is the lowest compared with starch or fat. However, there was no difference in ME to DE ratio between soybean expeller and corn. On the other hand, corn has the greatest NE to ME ratio compared with the soybean products, and this effect can be also explained because of the higher crude protein concentration in soybean products compared with corn. However, this observation is also telling us that the soybean expeller has a better utilization of energy compared with soybean meal. So now let's move on into the conclusion of this study. Based on the result of the study, soybean expeller had less crude protein concentration and a higher oil concentration compared with conventional soybean meal. By having the nutrient composition and based of the result of this experiment, we can conclude that soybean expeller has a greater standardized ileal digestibility of most of the amino acid compared with soybean meal, but the digestibility of fiber was not different between the two soybean products. In terms of energy concentration, metabolizable energy and net energy was greater in soybean expeller compared with soybean meal. And with that, I would like to thank Instapro International for the financial support and also all the members from the STY Monogastric Nutrition Laboratory. And if you want more information about all the work that is done in our lab, you can visit our website at nutrition.ansai.illinois.edu.